Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, Assistant Professor at Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. In our novel drug delivery systems, we started with a new topic, that is scalability and regulatory aspects of novel drug delivery systems. In our previous session, we have discussed the importance of novel drug delivery systems, how these novel drug delivery systems are different from the conventional formulation, and also we have seen some of the advantages of the novel drug delivery systems. Then we have discussed some of the marketed technologies, also known as the commercialized products, and then we have seen some of the products which are approved by the US FDA, that is US Food and Drug Administration, and also we have seen the from the Europe regulatory body EMA, European Medicines Agency. So, that suggests that there is a huge demand of converting, translating of the drugs as well as conventional drugs, conventional formulations into the novel drug delivery systems. And there is a huge requirement of NDDS for translation of such lab as well as R&D formulations into the clinic. So, also we discussed the challenges which are involved in the translation of the nanomedicines as well as novel drug delivery systems. So, today we are going to discuss the new topic in the scalability and regulatory aspects of drug delivery systems. Yes, as we discussed in our previous session, we end up with the challenges of the novel drug delivery systems. So, today we will focus on dedicated session for the current issues and the challenges of the novel drug delivery systems. So, here as we know that these few points we discussed in our early lectures, right. So, when you compared with the conventional, there are the cases you will see the improper drug loading with the within the formulations. There are the drugs which are water soluble and there are few drugs are water insoluble. Such cases, the selection of your material is very crucial. Based on the drug properties, we need to select right material. So, for that we need to go with like a specificity of the polymers or materials or lipids based on the drug properties. So, which that leads to the limited drug loading. That means, the after formulation you will see very less amount of drug in case of, best example in case of nano formulations. If you are taking in a hydrophobic type of the polymers or lipids, the chances of loading of water soluble drugs, especially in like a herbal drugs or natural active components, the percentage of loading is very less. So, that is one of the major issue. For that you need to understand the we need to optimize the formulation to improve the drug loading of the formulation. And the second thing is the burst release, which may happen at the target site, even with it which happens with the control release formulation, and whether various types of gastroretentive formulation, some case also happens with the nano formulation. So, the burst release that leads to the patient may undergo lot of complications because of the burst release. So, that need to be controlled and overcome by doing the proper formulation as well as evaluation studies. And the third current issues and challenges is material toxicity. So, here we are going to use various types of the polymers as well as various excipients which are works as stabilizers, as well as some of the preservatives we use it in a formulation. So, the material toxicity is one of the major problem in the novel drug delivery systems. We also use some lipids and even surfactant to make polymeric micelles as well as for the polymer nano formulation, even lipid nanoparticle. So, the toxicity of material is one of the major limitation, and yes, that need to be proven in the by doing of the studies, and after proving the safety only we can use such materials for the treatment. So, material toxicity need to be overcome, or we need to get the information of the toxicity of your material. And the fourth one is the cost is very high compared with the conventional system, because of you are going to use the dedicated instrument for each process. So, consider if you want to make a formulation with of nano suspension, you need a high pressure homogenizer if you want to do the scalability studies. Yes, we can also use some other technique like high speed homogenizer, again the cost goes down, but most of the techniques are not scalable. You can also do by other techniques, by using of ultrasonication also you can make nano suspension, but because of the regulatory and the scale up issue you cannot go ahead. But when you want to go, like for example hot melt extrusion is one of scalable method, spray drying also is a scalable method, microfluidics also one of the scalable method, but the cost of instrument is very high, and also it involves lot of steps from the raw material selection to the finished product. So, that will increase the cost as well as complexity in the formulation. So, that is also one of the major issue in the novel drug delivery system. And yes, stability issue is also one of the major problem. So, you will see the nano range or your polymer is coated on the surface of your carrier, it may be tablet or any formulations, but during the stability you will see the separation or the improper effectiveness of the materials. You will see some of the aggregations. Consider in case of nano suspension, you will apply high pressure through homogenizer or high pressure homogenization. Because of that high pressure and impact and shear, you will get a nano formulation after doing or stabilizing with the some surfactant. But during the storage, when you keep it in a different room temperatures or different conditions and temperature as well as relative humidity, what happen? The particle will start aggregate because of the high surface energy. That is a one more problem, and drug leakage which we already discussed in case of liposomes, and material degradation also one of the problem, like lipids undergo oxidation, undergo the degradation. So, that is why during the stability most of the formulations fails. So, we need to understand the stability studies and stability issues of the formulation, and we need to select storage conditions also. Packaging material also is very important while performing the stability studies. So, that need to be focused while developing the nano medicines or novel drug delivery systems. So, this is the one of the major problem, like batch to batch variability, because of the materials which are very sensitive and that undergo lot of variation and the alteration. Consider we are preparing a chitosan and the nanoparticles. You use chitosan as an polymer, right, which contains positive charge because of the amino groups. Now, we want to use some complexing agent, that is sodium tripolyphosphate, which provide the negative charge because of the phosphate group. Now, this amino groups and the phosphate group ratio is very important for the preparation of the nanoparticles by the complexation method, right. So, try to understand small proportion or variation in the chitosan and even in the TPP that leads to the alteration in the particle size, right, and also in the particle size that will also leads to the alteration in the PDI, polydispersal index, and same this charge is very important, that will also varies your surface charge of your formulation. Now, these are very crucial when I am working with the biological molecules like siRNA, again which contains the negative charge, and yes, I want to complex with my chitosan, proteins and peptides. So, try to understand the batch to batch variability is very high. Small minor change also will impact your outcome majorly, and reproduce also one of the major problem. Even though if you take like scalable techniques also, right, you will not get similar results from previous batch to present batch. Even though if you perform after 1 month with a new batch, you will do not see the reproducibility within the batches. So, that is why that is one of the major problem. Now, all the root cause here also one more problem, that is a scale up difficulties. Now, we can make a 100 nanometer formulations, maybe with the PLGA or chitosan, any type of formulation or solid lipid nanoparticle, but most of the methods are not scalable. Consider in case of we are preparing any formulation in the beaker, and by using a magnetic stirrer or by using simple technique, right. If that is not scalable, there is no use, and you have to control your process parameters and you need to optimize. Your method need to be scalable, and you need to understand all type of process parameters for that batch or for the technology. If you know that, you can translate or you can scale up your batch from the R&D formula to the large scale. So, the difficulty is also very high in case of novel drug delivery systems. Now, you can compare this with the conventional formulations. Yes, we can give high amount of drug as well as we can control this, right, and material toxicity is very less. Why? Because of all these are the GRAS materials, generally regarded as a safe materials we use. Of course, in nano also we use, but we are adding multiple ingredients that leads to the variations in their safety profile, and also which are biocompatible and which are already approved materials we use in the conventional other formulation. And manufacturing cost is because industry is well settled and they have the all type of the they have enough data on the various types of conventional formulation. For this nano, we do not have proper standards and the regulations. So, that is why. So, these are the some of the current challenges, and now further we can focus on few types of the challenges that can be classified into 4 categories: process related, quality related, and the regulatory and economy and supply chain challenges. We will see one by one and we try to understand and we will focus and we will correlate how these challenges will help us to improve the safety, efficacy as well as quality of the medicines in the commercialization. Now, we can see here the first point, the current issues and challenges. The first we are going to discuss on the process related challenges. As we discussed, reproducibility, scalability is one of the issues. Now, we know the preparation of liposomes we can make by using of thin film hydration method, which are often not scalable. Now, you can see here how we do. This is a rota, right, and in this what we do, we will take we dissolve the lipid in the organic solvent and then we evaporate it solvent and it will make a thin film. After that we hydrate and then we go for some sonication, right. So, from that you will get the liposomes. If there are the changes, like if you want to add some of the agents, you can add either this phase or this phase. So, you can add even drugs also, but in a general way you cannot translate this into the scalable method, right, because even though if you are making like a 5 liters or 10 liters, even 100 liters also, you do not have any critical process parameters for your formulation. So, this minor change will affect on your finished products and quality of the product, right. So, that is why these types of methods are not scalable. That is one of the main process challenge. Batch to batch variability is one of the major concern because of it depend on the manual step. Yes, we put the temperature as well as mixing time and the concentration of the molecules, right. So, which are completely manually and we will do with the hand shaking sometimes, and we use large rotary evaporators to remove the solvents, right, which are difficult to scale and control for the large scale production. And now, here you can see these are the physical and chemical conditions. Mixing time, temperature, concentration are differ significantly between small batches and large industrial scale batches. A small change in a stirring speed and the temperature can drastically change the size and morphology, drug loading and drug leakage. And you can read lot of papers variations within one batch. Even if you change small RPM, right, and also small temperature, that will affect lot on the particles and morphology, drug loading and drug leakage. These are the main physicochemical properties in the nano formulations. So, the batch scale or lab scale methods are one of the major process challenges. Second is, we are going to discuss on the some of the process challenges which are related to the product. If the carrier is degraded, for example, after preparing of your liposomes by scalable method and you have enough amount of drug loading, but still if your lipid layer undergoes any breakdown or degradation by some stability or in vivo or biological conditions due to interaction with surrounding environment or specific chemical agents, that leads to the premature release of the encapsulated drug before reaching the target site, then it will lose its effect. Now, after doing all this, if your carrier goes undergo any type of degradation, either it may be the polymer, lipid or any material, so then it will lose its purpose. So, we need to know before developing or translating of your formulation, we need to know what are the process challenges for your product. Now, nanoparticle aggregation, this may happen because of coalescence, even though because of the high surface area that leads to form the aggregates. So, nanoparticles are prone to aggregation where they clump together. This process can lead to larger particle that is not at all good for the in vivo or body in the body, that may be cleared from the body more quickly or fail to reach the intended target. Not only that, that will create a lot of difficulties in the patients, thereby diminishing the therapeutic benefit and potentially causing the toxicity in the body. So, yes, we need to avoid nanoparticle aggregation, and for that we need to understand different stability studies with the in use, and from that you can add various types of stabilizers, so that will avoid the all type of aggregations. Now, the second problem is quality challenge. Now, that is connected to the characterization. Now, characterization will give, after performing any characterization studies, you will understand whether your product is qualitative or not. We can decide the quality of the material after performing some evaluation or quality control or characterization studies. So, after doing the characterization, we will understand the Q, that is quality of the product. Now, try to understand this principle. You are putting X material and you are adding drug and you are adding some other stabilizers. Now, you are getting some product that is considered A, right. So, input, by doing the process you are getting output. So, now this is a black box, there we are unable to understand the interaction of X with Z and Z with Y and also X Y, right, and also the combinations of these three also need to be understand during the process. Now, by eliminating or by understanding of the interactions of the each material physicochemically as well as biologically, then that will give the right decision to translate the medicines. But in research we are so happy with the simple studies, and from that we will believe that we are providing a best formulations, right. So, but which are not up to the mark, that will not give any quality significance for your formulations. So, this black box need to be studied, and from that you will understand. Now, you can read here with me, while simple matrices like particle size, polydisperse index are more important, especially fields like nanoparticle formulation, they often insufficient on their own capture the full complexity of the system. So, getting 100 nanometer and the polydisperse is very less, like 0.1 is not give enough information, that is important, but beyond that we need to understand how the morphology, how the ratio of the materials is influencing the outcome of your material. And not only that, the output also we need to understand. A complete understanding requires multiple complementary techniques considering factors beyond the size and distribution, that means morphology, shape, kinetics, and analyzing the interaction between the components to understand the collective dynamics, right. So, dynamic properties of your formulation need to be understand, that will give the information on the quality. Now, here another one more parameter, that is characterization. Yes, we have nowadays the advanced techniques, that is dynamic light scattering, transmission electron microscopy, even zeta potential surface chemistry analysis. With these you will understand the realistic data of the nano formulation. Yes, that we need to more such type of techniques or standard characterization techniques is required to understand the morphology, shape of the formulations, and also interaction by doing of some dynamic studies. So, that will help us to understand the kinetic stability, release profile, and the behavior in the different environments. So, that will help. Now, we can see this simple carrier. you can see various shapes are available and also various types of the materials can be used that can be prevent aggregation, also help in the preparation. And now you can see here the surface properties as well as in the size. So, all these four have different applications or different outcome in the body. So, now here you can see this is the spectrum of cateration techniques. Now, you can see from here size, we have size we can perform by various instrument. You can go further, here is the open access paper, you can read it, and also there are the composition you can also understand by various analytical techniques, and that will help for the understanding of physicochemical properties, and also surface structures, size, charge, and also you can see the impurities also by using ATR, FTIR, LCMS and NMR and GCMS and by using of IR. And also you can see the sterility you can maintain, these are the tests will help to maintain the sterility, drug release, also stability and the morphology by using of these techniques. So, you can come up with inorganic or organic material, anything that can be analyzed by using this type of analytical techniques, which are very, very important. And again in the second quality challenges, so here is the immunogenicity and toxicity. Yes, so here we use lot of excipients which are approved. There are the materials which will show the unwanted immune response and also toxicity in the body, that also one of the major drawback. And the second concept is protein corona effect. After giving the formulation into the body, so the body fluids have a protein that will adsorb on the carrier. So, that is called as protein corona. Now, the protein after adsorption, the physicochemical properties of your carrier will change. It may undergo aggregation, it may be stabilized, size also may vary, surface charge. So, it will also alter all the physicochemical properties, that also changes the biodistribution and safety profile. So, before giving this formulation, we need to understand the protein corona effect. This is nowadays one of the interesting topic in the immunogenicity and toxicity issues. Now, third one is regulatory challenges, lack of the information or established guidelines in the regulatory, that is fails to develop clear guidelines fast enough for new and innovative technologies. So, now regulation comes, you can see standards, rules, requirement, laws and policy that need to be followed, then it is considered as a compliance. So, lack of established guidelines. So, with that we are failing so many molecules in the market. Complexity, because of we are using both drugs and devices like combination products. So, that is also we learned this all in the intrauterine devices, right. So, the complexity because of the lack of guidelines, that is also one of the major issue in the development of the novel drug delivery system. This is connected to the regulatory challenge. Now, economic and supply chain challenge is also one of the major problem, that is a fourth point, that is high material cost, already we discussed, because of here we use the lipids which are sterile and the cost, the process is very, very high, storage, maintenance and extraction is very high, that is why cost goes very high, and also instrument and infrastructure investment is very high, but we have to invest for one time, but still compared with the conventional equipments and here the cost of the instrument is very, very high, and you need to come up with the good manufacturing complaint clean rooms with for such instrument. And securing novel excipient, nothing but in our market we do not get similar materials from other suppliers. So, that is also one of the challenging, this we need to perform based on the custom design. So, that is also one of the major issue. Raw material consistency, like quality, consistency, purity of the raw material used in the nanomedicine production is vital, and yes, the supply chain also is one of the major problem in the current issues and the challenges of the novel drug delivery systems. So far we discussed total 4 types of the challenges. The first one is process related challenges, second one is quality related challenges, third one is the regulatory challenges, the fourth one is supply and economic challenges. So, these challenges are very important. So, based on that, while selecting of your materials, even in terms of the process as well as characterization techniques, and for the regulatory we need to consider all these points. This will help to overcome the all type of issues, challenges in the development of novel drug delivery systems. With this, I am going to stop the today's session. In our upcoming classes, we are going to discuss on scalability, regulatory aspects of the novel drug delivery systems. Thank you so much.